Hello everyone. Today we are going to discuss about the transformation in the chloroplast. How chloroplasts are transformed. As we know, the chloroplast is the organelle which are present in the plants. They are having their own genome. They are mostly involved in the photosynthesis as they have their own many copies of genome are present in the stroma of the chloroplast. So, due to this advantage, we are able to insert our gene of interest into the chloroplast. So, we are having many different crops which are successfully transformed through the chloroplast genetic through the transformation by the genetic engineering. Like we are having different crops cabbage, cauliflower, lettuce, rice, soybean, potato, tomato. But these crops are having we are using for these crops we are using different selectable markers along with the gene of interest. And the most important thing is there that there we are inserting our gene at a specific site, that is site of integration of the transgene is fixed in every crop plant. So if we will take the example of cabbage, so we are adding AADA gene, or we can add VIDA gene, and the site for this AADA gene or VID gene in the cabbage is TRN1, TRNV or RRN16S, or we can insert our these genes in cabbage in TRN1 or tRNA or RM2 RRN23S. So these are the different sites of integration in case of cabbage. For cauliflower, again we are having AADA gene, and the site for integration is ACCD or RBCL. While in case of lettuce, we are having AADA gene of interest, and we can insert it TRN1 or tRNA site. So we are having different sites for different crop plants. By using the genetic engineering in case of chloroplast, we are having unique advantages. Like we are having high level of transgene expression. Why we are having high level of transgene expression? Because the copy number of the genome in the chloroplast is very high. So we are able to insert multigenes. So multigene engineering is applied there. And as well as the most important thing is that in case of chloroplast, we are not facing the problem like gene silencing or the pleiotropic effect, or we can say that there is no undesirable foreign DNA. The insertion of gene in the chloroplast is stable. So these are the important advantages of the transformation of chloroplast. Due to this advantage, they have overall reduced the fermentation costs which are commercially available for these crops. Like we can say that the plants which are genetically modified or genetically engineered are the bioreactors. Due to these bioreactors, the cost effective solution for using plants are into the action. By this technology, by using genetic engineering in case of chloroplast, we have got many successful stories in case with related to the herbicide resistant, insect resistant, or it is in the case of the disease resistant, drought resistant, and as well as for the salt resistant. Moreover, we can say that all the transgene and the insertion of the transgene in the chloroplast are also involved in the phytoremediation. Even we are able to get many vaccine antigens, like we have seen in previous lecture, like edible vaccines, and these chloroplasts are able to form many biopharmaceutical proteins. So they are having, so there are multi uses there. So all the therapeutic proteins, all the vaccines, all the things are we are getting at the very low cost. We are getting at the very low cost. If we'll talk about the chloroplast transformation, whenever there is gene insertion into the chloroplast, this gene insertion is always carried out between the two functional gene, or we can say this integration of the transgene is carried in the spacer region of the two functional genes through the homologous recombination. So if there is the homologous recombination, then we can say we are inserting our gene at the specific site. So we can say site specific integration is there. So wherever we want to insert our gene, and we will be knowing how our transgene will be enhanced, there only we will be able to insert our genes. As we know that we are not facing any gene silencing in case of chloroplast transformation, and it has been observed that there is the high accumulation of foreign proteins, and it has been reached up to the 46%. Thousands of copies of chloroplast genome are present in single plant cell. So we can say the expression of multi-transgene as a operon, as we have studied the operon system in case of bacteria. So similar way we are able to insert our multigenes. They are translated, or they are getting transcribed into a single messenger RNA. Then again they are multigenes into a single transformation step. So there is no processing of the, or we can say there is no formation of the monocistrons. All the genes are transcribed and translated together. Foreign proteins synthesizes the foreign proteins which are synthesized in case of chloroplast have ability to fold properly. Means whenever we have to go for the like whenever the protein is formed, it should be folded in a proper way so that its activity is achieved. When there is no proper folding, then that protein is lost. So whenever it has been observed in case of chloroplast transformation, the chloroplasts are having ability to fold the foreign protein properly along with the appropriate post translational modifications, including which includes the disulfide bonds, or there is the addition of carbohydrates, or we can say there is the addition of lipids to these proteins. In general, we can say this chloroplast transformation technology has eliminated the fermentation technology, where we used to go for the downstream processing of our product. So whenever the proteins are formed in the plants, so these proteins are encapsulated with the plant cell wall. So we can say these proteins act as are biocapsulated proteins. Whenever we are taking these proteins, these are not digested by, or these are not digested by the stomach enzymes or acids, but in the gut of human beings, these proteins are able to get digested due to the presence of bacteria which are naturally occurring in our gut. So if we will talk about the chloroplast transformation, so we are taking the basic plasmid promoter, With the promoter, we are having flanking sequences and chloroplast specific expression. So we are taking that promoter only, which will be chloroplast specific along with the flanking sequences and selectable marker 5' and 3' untranslated regions to enhance the level of transcription and translation. So these regions are regulatory regions. So we are using different regulatory regions at the 5' end of the gene insertion and the 3' end of the transgene. For all these regulatory sequences has been derived from the tobacco plant itself, and the sequences or the genes which are used for 5' regulatory sequence are PBSA and RBCL genes, and for the 3' end they are, and for the 3' regulatory sequence they are PBSA, RBCL or RPS16 genes. So when if as we I just said that these sequences are derived from the tobacco plant, so these sequences are endogenous regulatory sequence of the plant. So the regulatory sequences are essential, or they play important role in the plastic transformation vector to compensate the degradation of the newly synthesized protein by chloroplast proteases. As there are many copies of the transgene are able to make the protein, so large number of proteins are there, so the cytosol proteases will not be able to break all the proteins, due to which there will be higher accumulation of the transgene protein into the chloroplast. Like we have taken example of heat shock proteins, and this type of example has been seen in the insecticidal proteins. In case of insecticidal proteins, Cry2AA2 folded properly when the chaperone was co-expressed. We had taken a gene from the Bacillus thuringiensis, which are against the insects. So the gene for that is the Cry2AA2. Along with these genes, we are able to go for the co-expression of the chaperones. So due to this chaperone, these proteins are not getting degraded and increasing the accumulation of foreign proteins. And it has been observed by insertion of the chaperones along with the Cry2AA2 gene, they have increased the accumulation up to 128 fold. Initially, they were having 0.36% accumulation of proteins in the chloroplast. Now, due to the insertion of the chaperone gene, now we are having 46 point higher accumulation in the chloroplast. So whenever we are inserting our gene, it should be inserted in a proper way and the integration should be very stable. So maximum, whenever it has been earlier the experiment were carried out, they have inserted the transgene at the spacer which is not transcriptionally active, so they observed that the protein expression is not high. For this reason, the maximum number of transgene has been integrated between the transcriptionally active site. Like example is TRN1 and tRNA. So we are having different 1,000 copies. So as chloroplast have ability to divide by own, so they are able to make double copies of their genome, due to which there is the high expression level, and higher expression is until now till date it has been observed the higher accumulation of foreign protein is up to 46%. Even for these, we have inserted the origin of replication into the vector, which are mostly meant for the chloroplast transformation. And it has been seen that integration of transgene between exons of tRNA and TR1. It has been observed that the integration of transgene between the exons of tRNA and TRN1 facilitates the correct processing of the transgene. So when we are inserting the transgene, so there must be some method by which we are able to deliver our DNA into the blasters. Mostly the widely earlier, or we can say that previously, we used to go for the polyethylene glycol mediator gene insertion, but nowadays we are having particle deliver system. Through particle deliver system, by which we are now able to insert our gene of interest by coating our gene with the tungsten or the gold particle and delivered into the plant cell using the particle bombardment. So by this way we are able to insert the gene of interest into the chloroplast. So here is a typical structure of tobacco chloroplast transformation. So in first diagram we are having restriction sites. This is artificially made vector, and we are having untransformed 41.1 kilobase pair. So this part has been removed, like we can see in the diagram B. We have inserted within in the same sequences SMA1 restriction sites on the both 3' and the 5', and we have inserted their gene which is transformed, and the size of this transformed gene is 5.23 kilobase pairs. 5.23 kilobase pair, where we have inserted our, as I said, at 5' and we have to insert the regulatory sequence. It's 16S tRNL, then the promoter, then the ribosome binding site, then the 3' UTR promoter, gene of interest GOI, that is 2.25 kilobase pairs. Then again the 3' untranslated regions, that is 3' UTR regions. Then again the regulatory sequence which are present at the 3' end, that is tRNA23S. This is how the vectors are constructed for the chloroplast transformation.